Titer anti-HIV antibodies are common even in healthy individuals. High-titer anti-HIV antibodies are pathognomonic in chronically active hepatitis. The anti-HIV test does not answer the question whether anti-HIV antibodies occur or not. The test differentiates between plenty equals positive and few equals negative. Rethinking as to reverse transcription. The error taking proteins resulting from HIV isolation for retroviral proteins dates back to 1970. The paradigm that DNA codes information and programs relating to all physiological and phenomenological aspects of all organisms resulted in the postulation of the irreversibility of the genetic flow of information for the synthesis of proteins, from DNA via messenger substance, RNA, to proteins. This was the crucial genetic dogma. 25. Despite the proven reversibility in 1970 that from RNA DNA can remerge, this fact was postulated as an exception that proves the rule, by stating the existence of retroviruses qualified for this reversibility, which at this time were only considered as tumor viruses. 26. 27. With the discovery of this enzymatic activity in all living cells, it soon became clear that the evidence of the function of reverse transcription from DNA into RNA was not a proof for the existence of retroviruses, because the genome of all eukaryotic cells is clearly marked by this activity. 28. 29. Retrospectively, it seems rather astonishing that in 1983 Montagnier and in 1984 Gallo still postulated a new retrovirus despite the fact that a new viral entity had never been isolated or described, according to the standard regulations in virology. As a matter of fact, the enzyme reverse transcriptase from a HIBL has never been isolated or described, but only inferred from functions to its existence, when new formation from DNA into RNA was proven by laboratory techniques. Since 1985 it has been known that the reverse transcription plays a decisive role in the maintenance of the structure of the genome, by repairing chromosome fractures and especially by limiting the loss of chromosomal end components, the telomeres occurring at cell replication 30 to 33. The respective enzymes for this kind of reverse transcription, the telomerases, dispose of a type-specific RNA matrix for the formation of the repeating telomere units. Somatic human cells that do not belong to the reproductive path cannot adapt to the shrinking of their telomeres when replicating, and so cease the replication at a certain degree of depletion. Up to date, the influence of nucleoside analogues on the action of telomeres at replication has obviously not been investigated. We could trace only two publications, in 1996, which describe an in vitro investigation of nucleoside analogues inhibiting the telomerase activity. In our view, the knowledge, already gained in 1980 on the vital physiological function of reverse transcription, should have led to a rethinking as to the establishing of nucleoside analogues as pharmacological inhibitors of the reverse transcription of the postulated high viruses, and, according to knowledge of that time on the physiological function of the reverse transcription, it should have been rejected. 34,35. What is the decrease of CD4 lymphocytes in AIDS based on? The decrease of the circulating CD4 lymphocytes in the bloodstream during progressive immune deficiency in AIDS has generally been explained by the progressive destruction caused by high viruses. 36. 4 years ago Carbonari et al. Showed in an in vitro investigation that the apoptosis of the lymphocytes in AIDS patients is mainly related to CD8 T cells and CD19 B cells. 37. Finkel et al. Then pointed out that apoptosis concerns mainly bystander cells, and spares supposedly infected cells from so-called HIV and SIV lymph nodes. 38. These reports remind us of Fossey's classical publications of the 70s, in which he and his working group clearly demonstrated that, in persisting hypercortisolism, an increasing number of CD4 cells leave the bloodstream, and can thus activate B cells in the marrow. 39 to 44. The migrated CD4 cells return to the bloodstream upon dropping to normal values of the cortisol level. At the beginning of 1995, Wei and Ho et al. published a report in which they declared that the extremely fast multiplication of Hi-1 viruses produces a raised turnover of CD4 lymphocytes, 45-46. Towards the end of 1996, Walters et al. Showed that the telomere length of CD4 lymphocytes in anti-HIV positive individuals remains normal, whereas the one of CD8 cells decreases, 47. During the latest International Congress of Leading HIV Scientists, the long-term criticism of the HIV/AIDS theory has been confirmed. Despite intense and precise investigations, there was no proof of a pathophysiological mechanism explaining the different reaction of CD4 and CD8 lymphocytes to the postulated retrovirus HIV. 48. It was literally stated, the riddle of CD4 cell loss remains unresolved. Paul Johnson of the Harvard Medical School in Boston voiced in a disillusioned way the helplessness of the conventional AIDS scientists. We are still very confused about the mechanism that leads to CD4 depletion, but at least now we are confused at a higher level of understanding. In other words, Fauci's pioneering work, based in the 70s, on experimental traumatology fell into oblivion. Calvano clearly documented in a review published in 1986, that the selective depletion of CD4 lymphocytes is induced by neuroendocrine mechanisms in traumatic conditions such as injuries and burns, whereas its proportions depend on the degree of hypercortisolism. 49. It remains enigmatic why Fauci, after having joined the AIDS research, never again mentioned his own reports. What does the viral load measure? Immediately after the publication of Ways and Ho's reports in January 1995, 45-46, in which they put forward the hypothesis that Hi viruses multiply at raving speed, destroying a similar number of CD4 helper cells, quantitative tests based on the genetic multiplication method PCR were introduced, and so a large number of HIV in the bloodstream was postulated. It has been well known among HIV scientists that the so-called viral load, i.e. the measurement of the viral load, is no evidence of the entire virus genome or intact viruses. 50. The viral load measures short components of the messenger substance RNA, attributed to the Hi viruses. Because the Hi virus genome per se never could be described, it is impossible to designate these RNA fragments as viral. Going through the records of the presumed characterization of HIV, it can be inferred that all components, proteins and genetic substance attributed to HIV are of pure cellular origin. 3-7. Therefore, the results of the viral load can only have an indirect significance, such as the measurement of an increase or decrease of cellular RNA, as it can be observed as increasing in catabolic conditions of cell disintegration and decreasing in anabolic conditions. However, these results cannot be considered as clinically relevant as, besides the technical inadequacy, control investigations with both non-positive defined healthy and ill individuals have never been published. The polymerase chain reaction (PCR) is a technique for a manifold multiplication of short DNA fragments, developed by Nobel Prize winner for chemistry, Kary Mullis. Upon measuring the viral load, the RNA fragments in the bloodstream first have to be converted into DNA, and then multiplied as such. The single, developing technical steps of this method are prone to failure. The slightest impurity, drugs, such as heparin and other substances, interfere with the reproducible functioning of the PCR method, especially with quantification. 53. Kary Mullis, the inventor of this method, does not miss any occasion to criticize the application of his technology in the context of AIDS. 52. Furthermore, it is concealed that it does not make sense, either practically nor theoretically, to initially multiply manifold fragments of genetic structure and then to postulate their manifold presence. In case they were actually present in the blood samples, it would cause no problem to prove their existence by simple, quick, and cheap standard methods. 51. And if de facto viruses did exist in the bloodstream, scientists certainly would have been successful in making them visible. Hence, up to date, no scientist claims this achievement, a fact which has been confirmed under coercive evidence by the German Health Ministry in 1996. After the report of a produced positivity in the viral load during a vaccination test with proteins, 54. Of a previously negative defined test person, it is now frankly admitted that repeated wrong positive results in a viral load are quite a well-known phenomenon. 55. Impairment of energy formation in mitochondria by nucleoside analogs such as AST, azithromycin, zidovudine. AIDS patients quite often demonstrate a weakening of their skeletal muscles. Up to 1990, this was considered a high virus-caused impairment of muscles. In 1990, Delacaz et al. Demonstrated that this kind of muscle disease is due to an administration of AST, weakening the mitochondria within muscle cells. With the excessive release of free radicals, the mitochondria are affected in their function of forming ATP as key substance in metabolic energy. 56. Hayakawa